Hello, everyone. Welcome to the Project Tenure Spring 2020 webcast series with today's speaker, Keith Baggerly. I'm Jenna Kroll, Assistant Professor of Biostatistics in the Department of Global and Community Health at George Mason University. I'd like to begin by thanking co-directors of Project Tenure, Norm Medeiros and Richard Ball, the Project Tenure Executive Committee, Alex Savith, as well as the generous support from the Alfred P. Sloan Foundation for making the Project Tenure webcast series possible. Today's webcast will be recorded and posted to the Project Tenure website. Now it is an absolute pleasure to introduce our speaker, Keith Baggerly. Dr. Baggerly was a Ransom Horn Jr. Professor of Cancer Research at the University of Texas, MD Anderson Cancer Center until his retirement in 2018. He received his undergraduate and graduate degrees in statistics from Rice University. Dr. Baggerly is an innovator and leader in the field of reproducibility in biostatistics and bioinformatics. His work has been highly impactful. It has led to new guidance by the FDA, an Institute of Medicine report on how omics assays should be used in guiding patient care, and new guidelines from the National Cancer Institute, as well as it helped prompt NIH's 2016 Rigor and Reproducibility Initiative. Personally, for me, it is so exciting to be introducing Dr. Baggerly, as I have used his work featured in the New York Times as well as 60 Minutes to teach reproducibility in my own courses. Today, Dr. Baggerly will be presenting his talk, The Importance of Reproducibility in High-Throughput Biology, Case Studies in Forensic Bioinformatics. This talk will discuss the approaches that must be taken to reproduce results when documentation of methods is inadequate and how such approaches can uncover errors. This presentation will last for approximately 45 minutes, after which we will have some time available for questions. Please type your questions into the Q&A panel on Zoom. I'll assemble and read the questions as time permits at the end of the talk. Thank you so much for attending today, and I will now turn it over to Dr. Baggerly.
Okay, thank you very much. Let's see. Okay. I believe I am sharing my screen now. Am I correct? Let me see. System preferences. My apologies. So I am told that Zoom will not let me share my thing until I have quit it and restart. Oh. So I believe I'm going to have to bow out and reappear momentarily. This is not something I was expecting, but what the heck. Welcome to the world of weirdness. So I will be right back.
We will wait here for you, and hopefully you will return shortly.
That is my plan. Okay.
Wonderful, we can see you perfectly.
That looks much better. And so, hello everyone. Even if I cannot see you, I appreciate your showing up and saying, I want to spend my afternoon with the statistician listening to the importance of medical data, because that might be appropriate at this time and place. Now, just as some background, I worked at the MD Anderson Cancer Center for about 20 years. And when I was there, I was working as a member of the Department of Bioinformatics. And bioinformatics at MD Anderson is focused on, I will term it, machine learning or data and data diving or whatever. The idea is we're now at the point in time where we can measure lots of things about patients as they come in the door. And we're trying to figure out, of those lots of things that we measure, which of them are important for guiding patient care. And some of these may be important in ways that we hadn't expected, so we're going to try to figure that out. And the thing is, as we started doing this, and as we did this for the first few years, we explored new types of assays, new types of ways of measuring protein or RNA or various genes and whatever. And one thing we discovered fairly early on is that while we can measure tens or thousands of things for each patient or each patient's tumor or whatever, our intuition about what makes sense is very poor when we start dealing with that many things. And what I mean by that is that there have been times when I've worked with a clinician, I said, hey, this gene appears to be overexpressed, and the patients do worse. And I've had the clinician say, well, you need to go back and check the data because we know clinically that it doesn't work that way. They've had that background intuition and they know how that works. But the problem is that if I go to that same clinician and I say, wow, I've been looking at this set of 100 genes and here's this pattern. And if I see this pattern, the patients do well or they do poorly. And it's a wonderful experience because it's sort of like conducting a Rorschach test with biologists, because you can see them go, oh yeah, I can see how that works. This interacts with this, this interacts with this. And you can see them construct a story right in front of your very eyes. And we know, unfortunately, that a few times that story is wrong. And what that means is that we're faced with a dilemma. We really want to be able to use the patterns that we find in high dimensional data to improve patient care or do other things. But if we can't trust our intuition, how can we trust this whole system? Well, it turns out that the way that we figured out, or the way that we decided we needed to do it, was we would be okay trusting the system if we knew how to trust each of the steps that we used to build up our patterns very well. And so that's what led to reproducibility and tracking each of those steps. The problem that we then encountered is that when we went over to the literature and tried to say, this is a wonderful paper, let's take this, let's carry it forward and let's apply it, is that we discovered that in many papers, the amount of detail supplied is not enough for us to figure out or precisely replicate or reproduce what they say they've done. And what this led us to do several years ago was actually to develop a brand new subdiscipline. And we call this forensic bioinformatics. And quite frankly, what this is the art of saying, okay, here's the data they started with, here's the results they reported, what the hell must they have done to get from one place to the other? So this is reverse engineering by another name. And this, we have found, is often informative, though not always in the way that one might expect, but it's actually something that turns out to be important. And I'm going to tell you today about a particular case study where we had to apply these methods to deal with a clinical question that's of real interest, specifically, which cancer patients will respond to which chemo agents. This is clinically important because if a woman shows up at MD Anderson today with advanced ovarian cancer, we will give her standard frontline therapy, which is a platinum agent and a taxane. And we will do this knowing that for about 75 or 80% of the people we give this treatment to, it will help. The tumors will shrink, the patients will feel better, we will have improved their quality of life. The other 20% we will poison for about 6 weeks to no good effect. We will do this because we don't know which 20% they are going in. If we knew which ones they were, we would try something different. And it is against this backdrop that a paper appeared several years ago in Nature Medicine that got a whole bunch of people, including several teams at MD Anderson, extremely excited because the authors claimed to have largely solved this problem. They could figure out which drugs would work in which patients. So here's what they said they could do. What we're going to do is we're going to start with new sets and new assays that can measure thousands of things, specifically microarrays. These are measuring RNA levels of thousands of genes all at once. And what they did is they said, all right, we're going to start with a bunch of tumor samples, but rather than starting with patient samples, we're actually going to start with samples from a bunch of cell lines, things that we can grow in the lab. And in particular, we're going to start with a panel of cell lines maintained by the National Cancer Institute for about the past 40 years called the NCI 60. Quick quiz for the audience. Everybody realizes that the number of cell lines in the NCI 60 is 59, you got that right? Okay, checking data is important here. The fun thing is that they then said for all of these cell lines, what we're going to do is we're going to assay them with these microarrays, and we're going to get thousands of measurements for each cell line of all these genes. And then they said, all right, what else do we know about these cell lines? Well, one of the reasons they're using this panel, one of the reasons the NCI has maintained this for a long time, is that every time a new chemo drug is introduced, the NCI tests that drug for how effective it is against every member of this panel. Specifically, if we come in with a new drug, they will say, okay, what is the concentration of that drug required to cut the growth rate of this cell line in half? And that information about how sensitive each cell line is, all of that is publicly available data. So for any given drug, we know which cell lines are most sensitive and most resistant. So the fun thing is that if we can leverage that type of information to say something about here's how cell lines respond and say something about here's how patients will respond, we could then use this in this set of data that we already have as something like a Rosetta Stone to determine that type of pattern for every drug we have. So here's what they did. They said, let's start with a drug of interest. Let's say docetaxel. We're going to go to our cell lines. We're going to sort them by sensitivity to docetaxel. And having sorted them, we will take the most extreme ones, let's say 7 cell lines at each end. And then we're going to compare those cell lines and their microarray profiles gene by gene, one at a time, using immensely sophisticated mathematics like 2-sample t-tests. And we will identify the top 50 or so genes, the most different ones, and we'll say the pattern of expression in these 50 genes, that is the signature of sensitivity to docetaxel. If we look at the microarray profile from a patient sample and we compare it with the samples from these various cell lines for these 50 genes, if it looks more like a sensitive sample, we'll give that patient docetaxel. If it looks more like the resistant samples, we'll try a different drug. And the reason this got a bunch of people really excited is they reported trying this for 7 widely used drugs, and they reported getting great results every single time. Now, what this meant is that within weeks of this paper appearing, we had a whole bunch of different clinical groups at MD Anderson coming to us in the bioinformatics department saying, we want to do this. We want to use this method. We want to help our patients do better. This would be a great thing to use. But the method uses math, and that's very scary. So can you help us? And we said, okay, great, sure, we'd love to do this, and we'd love to try. And the thing that made us really excited about this is that the data that they're using in this paper, that they're using to make all this claim, every single bit of the data that they're using is publicly available. So we thought we should be able to go in and test it and make sure that we knew precisely what was being done and we could build on it. So we said, that looks really cool. So we decided we're going to try to do this ourselves. And we decided that we were going to start by trying to do something easy. Specifically, we said, let's take one of the drugs they looked at, again, let's say docetaxel, and we'll see, can we identify, when we identify our top 50 top genes and we compare them with their list of top genes, what's the overlap? Because we'd expect that to be high if we're doing it right. So we tried this, and we took, say, 5-fluorouracil, one of the drugs, they reported 45 genes. I did the 2-sample t-test myself. I'm a PhD level statistician, I can do this. And I looked at the top 45 I got and the top 45 they reported, and the overlap was 0. And we said, that's probably bad. So what the heck was going on? Well, one of the things that it turns out is important is that when you start dealing with assays or measurements of thousands of things, at some point you stop referring to them by name. You start using various indices or various things that can help the computer find them. The reason this is important is that the assays they were using, the microarrays, were made by a company called Affymetrix, and each of the genes, probe sets, was assigned an Affymetrix probe set ID. Now, the way this shows up is that if we look at the list of the genes that they say are important, here are the probe set IDs that they identify as being really, really important. Here are the probe set IDs that we identified as being really important using 2-sample t-tests. And the fun part comes when you start looking at their list and then going over to ours. 1, 2, 3. And several of you have probably noticed the pattern here. We have an off by 1 indexing error. So what has happened? Well, we managed to track down the software that they were using, and because there wasn't any documentation for it, I wrote it. And in the process, we figured out how their software works. Their software, as it happens, works by taking 2 inputs to produce the results. The first input is a big expression matrix, a big matrix of numbers that's genes by samples, and there's a header row saying for each sample, is this sample sensitive to treatment or resistant to treatment? That's the first input. The second input is a list of gene names in the same order as the expression matrix, but, and here's the kicker, for their software to work correctly, this list of gene names must not have a header row when you input it into the software. So guess what happens if you're working in Excel and you cut and you paste and you leave on that one header row? You get an off by 1 indexing error. Now, what does this mean? It means that they were able to tell a story about why all of these genes were really important to how patients responded to a given drug, when actually none of those genes were involved in the biology at all. And they were able, that got by the authors, it got by the reviewers, it got by the editors, because, as noted, when we're starting to work with hundreds of things, not just 5 or 10, our intuition about what makes sense sucks. And we know this because this happened not just with this one drug, but it's actually a problem with all 7 drugs they looked at. In every single case, they got an off by 1 error, and they were still able to come up with a story that sounded plausible. Now, that was disturbing, but as we stared at it for a while, one of the things that came up was we said, you know, well, this is embarrassing, but it's actually not fatal for what we want to do. What we're hoping to be able to do is trying to predict patient response on the basis of what the gene measurements are. Those are the numbers. So if there are still patterns in the numbers that will tell us whether a patient gets well or not, we can still use this. Their labeling of the genes at the end of the day, that was broken, but if the numbers still tell a story, we can still use this. So we said, all right, since the people here are still excited, let's go ahead and take a look just at the numbers and see, can we make good predictions from the numbers alone? But that begs a different question, which is what level of prediction accuracy is good? So we decided that we were going to set ourselves a specific bar. Namely, we were going to say that if we could get predictions that were as good as the ones they claimed in their initial paper, then we thought we would have a good deal because, hey, it was good enough to get them into Nature Medicine. So with that in mind, let's take a look at one of their plots. And part of the question here is, how good are their actual predictions? Well, here, this is an example where they're looking at a cohort of 24 women who were treated in the Houston area, as it happens, with for breast cancer with single agent docetaxel. Most of the patients who responded to treatment, they were sensitive to treatment. These are blue squares. Most of them had low scores, that's good. And most of the patients who were resistant, red triangles, most of them had high scores. That's good, because there's clear separability between the 2 groups. They didn't get it perfect. They didn't get all of them right, but they got most of them right, and that's not bad. But here's a deep analytical question for you, and I'm a statistician, so I'm going to ask you to take a look at the data with me. And my question for you, and I'm going to give you a second or two to think about this, is how many blue squares are there in this plot? I'm going to give you a second here. When we counted, we saw 13 blue squares and 11 red triangles. That means that they're predicting accurately the outcomes for 13 sensitive and 11 resistant patients. The problem that we ran into is that I mentioned earlier that all the data that they're working with was publicly available. So in this case, we actually went back to the source paper for the data set they were looking at here. That source paper had shown up in the Lancet a few years before. And as we looked at that paper, the Lancet paper said, well, we're going to spend a lot of time talking about how to separate our 11 sensitive from 13 resistant patients. 11 and 13 not 13 and 11. And that alarmed us a bit. We said, okay, that doesn't sound right. Let's try another drug. And for those of you who are really ambitious, you can try counting the number of red triangles really fast. But I think you'll give me that there's a whole bunch more red triangles than there are blue squares. Again, they didn't get perfect accuracy, but they can say, we're predicting it pretty well for a drug which most of the patients are resistant to. Problem. The problem here is that this data came from a paper that showed up in the New England Journal of Medicine a few years before that. And the paper in question was dealing with the treatment of childhood ALL. It's a form of leukemia. The thing is, as cancers go, ALL is one we know how to treat. Our cure rates are about 80%. Now, what this means is that if in this patient population, we have a drug that most of the patients are resistant to, we wouldn't be using it. So this can't be right. And indeed, the authors of the New England Journal paper thought most of their patients were sensitive, not resistant. What's going on? Well, we went back to their software again. I told you earlier that their expression matrix had a header row saying, is it sensitive or resistant? That's not quite right. What they actually put in or supplied was a header row that had either a 0 or a 1 for each column, but they never explicitly wrote down 0 equals sensitive, 1 equals resistant. So in some cases, as they went from one drug to the next, they got the labels reversed. Now, what does this mean clinically? It means that if we use their method and their method actually works, it would recommend administering adriamycin just to the patients who are most resistant to it. And this is a bad idea. This is poor clinical practice. So we said, all right, we don't like that. So we went back and forth and tried it, and we communicated with the authors about this. And something to note is that this explanation that I've just given you about what we found, this was not the result of just a few minutes of looking at the data. This was actually the result of about 6 months of back and forth between us and the initial authors saying, guys, we'd really love to use your method. We don't understand it. Can you help us? Guys, we think some of the labels are off. And then eventually it led to, guys, if we use the labels that we think are right, we can't get it to work at all. Can you help us? But at the end of this, they basically said, well, obviously this interaction has ceased to be productive, so we want to bring it to a close. And we said, okay. And we wrote up our various objections and summarized the problems we'd seen, and we submitted that as a letter to Nature Medicine. And the following year, Nature Medicine published our critique. And when they did, they also published something else. They published the author's response. And we were really curious because we didn't know what the authors were going to say. We weren't sure if they were going to say, oh, we screwed up, we're sorry, we'll never do it again. But when they responded, that wasn't quite the tone of the response. The authors said, well, actually, unfortunately, the analyses by Drs. Baggerly and Coombs are deeply flawed. They didn't use our methods properly. And we have gone back and we have tried applying our methods to these data sets again. No, we've got our labels right. In particular for adriamycin, that's the one for all the red triangles. We're certain our data are correct. We have posted our raw data to the website to show this. And furthermore, in the interim, between the time our initial paper showed up and this paper showed up, we have continued to apply our method, getting great results every single time in other high-profile papers. So they must be wrong and we must be right. And we looked at that and we said, well, maybe we screwed up. Well, they said that they've posted the raw data for adriamycin, that's with all the ones with red triangles, to show that we got it wrong. Let's look at the data. So we downloaded the data from their website. It was a big Excel table, 144 columns with 2 headers for each column. And the 2 headers said, is this a cell line used for training or is this a patient sample used for prediction? And in it, the second header said, is it sensitive to treatment or resistant? And when we counted the number of patient samples that were labeled resistant, the number of red triangles, it matched what they'd found initially. So, well, that's weird. Okay, but we've now got the numbers. Let's at least look for patterns and see if we can figure something out as well. So as we looked for patterns, one of the things that we tried was for every column of numbers, we said, how closely linked is it? How tightly correlated is it with every other sample? And we plotted those correlations, those associations. And what I'm showing you here are just the cases where those correlations are very high, almost perfect. Now down here in the lower left, these are the correlations between pairs of cell lines. And this line of red dots on the main diagonal, what that says is that cell lines are highly correlated with themselves, which I believe that's correct, that's good. Now, but when we get to the patient samples, we see this line on the main diagonal, which we expect, but we see all of these other red dots, which we didn't. So what's going on? When we went back to their table and we looked more closely at the numbers they were using, we saw, oh, well, they've got 122 columns that are labeled as being patient samples. That's true. But some of those columns of numbers have been reused. This column actually has the exact same numbers as this one. So some patient samples have been reused 2, 3, or even 4 times. And right about here, there's one patient whose data was used 4 times, where that patient was labeled resistant 3 times out of 4. So we're pretty sure they don't have the labels right yet. So we wrote to Nature Medicine about this and said, guys, this doesn't work. And Nature Medicine said, well, the authors will make a correction. There's no need to publish your objection. And indeed, a year later, the authors published a correction where they said they reanalyzed the data using only the 95 unique samples, and they still claimed great results. The thing that surprised us was that when we actually counted the number of unique samples, we thought they had 84, not 95, so that was news to us. And when we looked at the 95 they said they were using now, it turns out that of the 95 they're using now, only 80 of them are distinct. 15 samples were analyzed twice, and of the 15 samples analyzed twice, 6 of them were labeled resistant one time and sensitive the other. So the correction of a correction still needed correcting. But Nature Medicine had said it was dead, so we moved on. We moved on in this case to one of the other papers that they said proved that they carried it forward that showed that it could work. This was a paper that showed up in the Journal of Clinical Oncology, where what they did this time was they extended the approach to dealing with 2 new drugs, 2 ones they hadn't looked at before, specifically cisplatin and pemetrexed. And they said, we can now get great predictions here. And furthermore, these signatures we get look plausible, because if we look in particular at our signature for cisplatin, we see that some of the genes we identify are ERCC1, ERCC4, and a bunch of DNA repair genes. And we know from looking at papers in the literature that these genes are specifically involved in the biology of how cisplatin acts in the body. So people in lung cancer at MD Anderson said, this is great, can you check this? So we went in and we looked at the data and we tried to check it ourselves. And working with pemetrexed, they reported 80 genes. We are able to perfectly match the 80 genes they report after we account for the off by 1 indexing error that they're still making. For cisplatin, they reported 45 genes. We are able, after accounting for the off by 1 indexing error, to match 41 of the 45. For those of you in the audience who are good at subtraction, that still leaves 4. So there's a question of what 4 can't we match? What 4 just don't come out of the data no matter which way we slice it? Well, here are the probe set IDs and the genes they map to that we can't match. One's for ERCC1, one's for ERCC4. Here's another one for ERCC1 and another one for FANCM, which is DNA repair. These are the genes that are cited to establish plausibility. And there's no way they come out of the data alone. Something's weird. Furthermore, even in this context, even given that we can't explain them, these last 2 are special. What makes these last 2 special is that those last 2 were physically never measured at all. What's going on here is that the genome has a lot of genes, and at the time these assays were run, there were too many genes to all fit on one piece of assay material. So the company Affymetrix split the genome across 2 different chips. The U133A had half of them, including most of the known ones, and the U133B had the other half. Since most people only cared about the known ones, they, and the authors here, only ran the U133As, but 2 of the best genes they report are on the U133B. So 2 of their best genes they've never measured. We thought this was neat, so we reported this to the Journal of Clinical Oncology, and JCO got back to us and said, we're sorry, we can't publish this. We sent them a second note saying, can you tell us why? And they sent us a second note saying, we're sorry, we can't publish this. So we moved on. We moved on to the other paper that they said validated their approach, and it's there, right there in the signature, saying validation. And what they did here was they extended the approach to deal not with one drug at a time, but rather to deal with combination chemotherapies. And again, this is really important in cancer because we don't try to hit a tumor just one way at a time. We try to hit it from several different angles in the hopes that something will work. And they claimed that if we use their approach, to allocate patients to treatment arms, as opposed to just doing randomization, which is what was done, we would have actually improved the response rate in this particular trial from 44% to 70%. That's a big deal. So we said, all right, can we understand what they're doing? Here, they're actually looking at two combinations. One is fluorouracil, epirubicin, and cyclophosphamide. That's a 3-drug combo. And the other is taxotere, followed by epirubicin and taxotere. The T here is the same, so this is actually just a 2-drug combo. So we said, well, let's take a look at the raw data, and it's available on the web, so we downloaded it. And we said, if we cluster the data, do we see 2 clusters, those who respond to FEC and those who respond to TET? And when we looked at the data, we didn't see 2 clusters, we saw 3. And we said, that's strange, why do we see 3? Well, we looked at the data more closely. And one of the things that we noticed is that the particular type of data they were collecting, known as Affymetric cell files, these cell files have information in them. First off, they have this, for each gene, they have the expression value, but they also have metadata at the top of each one, including some rows that say, by the way, when was this assay run? So on the right, we've taken these same samples and we've plotted them as a function of run date. This first block over here corresponds to this block. The black circle says that these are patients who were treated with FEC. That's one of the treatments. Green X's, those samples were excluded from the final analysis for some reason or another. This second block, that's the other patients who were treated with FEC. This third block, those are all of the patients who were treated with TET. What we're seeing here is a case of complete confounding of the experimental design. All of the patients who received one of the treatments, TET, all of their samples were run several months after all of the other samples. That allows for the possibility that something can distort those measurements that isn't the biology of interest. Furthermore, not only were they run several months later, they were run in a different lab, and as it happens, in a different country. So we're pretty certain that some of the distinguishing features they're seeing here are not due to biology, they're due to experimental artifact. So that's a problem. So we said, okay, we're not happy with that, but we'll still try to leverage things. And one of the things that we were also going to try to leverage is saying, okay, can we leverage at least the rules? In other words, we sort of understood on a one gene at a time level how they were combining results. How did they combine, or one treatment at a time, how did they put together the results of multiple treatments to get a single number out? So to answer this, we went back to their Nature Medicine paper, which said, here's in outline how we might do it. And they sort of outlined it with a 4-drug combo. And we looked at this new paper that has a 2-drug combo and a 3-drug combo. And we said, can we figure out the rules that they are using? So let's show you the rules. And this is the only slide with equations on it. I'm going to tell you that right now. So here is the first rule. The probability that a patient will respond to treatment with this 4-drug combo, TFAC, is given by the probability that they will respond to drug T plus the probability they will respond to drug F plus P of A plus P of C minus the product of all 4. Now, if there are a few statisticians in the audience, and maybe there are, I'm not really sure, you're probably going, wait, something's not right. And indeed, the authors knew that something wasn't right because all of the probabilities they got using this rule were bigger than 1. And they knew, well, okay, that's not right. They said, well, we know the biggest one should be 1, the smallest one should be 0, so we'll fix those and we'll linearly interpolate everything else in the middle. This step did not make it into the methods section of the paper. This is something we found out by tracking one of the Excel files that was floating around, mousing over the appropriate cell and saying, oh, that's what they did. But that was confusing. So we weren't too happy with that 4-drug rule. But what did they do in this new paper which validated things? Well, this is a 2-drug combo, and for 2 drugs, this rule should be mathematically valid. They just generalized it wrong. So the actual rule they used for 2 drugs, well, here it is. This does give you numbers between 0 and 1. I'll give you that. But the rule is different. And that's strange. It shouldn't happen that way. What about the 3-drug combo? Here's the rule. I have a standing offer to buy dinner for the first person who can explain to me where the hell this rule comes from. I have no clue. I can simply tell you it's what they used. But what I can tell you is that every single validation rule used is different. And as an American, I will tell you that I have a dream. And my dream is that if you're going to validate an approach in new data, that you would keep that approach the same. I don't think this is an unreasonable dream, but it wasn't the case here. So we said, all right, we're not happy with the design, we're not happy with the rules. Is there something else we can use? Well, maybe if we just look at it one treatment at a time, we can still get something useful. So we tried that. And indeed, in the paper, they said, if you want to predict our patient's response using just one drug, the single best way you can do that is to look at Cytoxan and use the values of that one gene, or for that one treatment, to predict response to FEC, that 3-drug combo. And this is an ROC curve, receiver operating characteristic curve, and this is a beautiful one. It says basically that using the appropriately chosen cut point, we can often almost perfectly classify the responders in patients into those who responded and those who didn't, because this curve goes almost all the way up to this corner. That's what they reported in their paper. When we tried fitting the data ourselves, this is the curve we came up with, which looks quite different. And then we stared at that and said, is there something in the numbers that we could try differently? And we stared and we stared, and then we tried something else, which is we looked at it and we thought, we asked a different question, which is simply, does Cytoxan make sense? And here, what had happened was we went over and we talked with some of our colleagues who are clinicians, and we asked them to tell us more about Cytoxan. And one of the things they told us is that Cytoxan is what's known as a prodrug. It's something that isn't active in and of itself. It's something that you give it to the patient and the patient processes it in their liver, and what is produced by the liver is the drug that actually works in the body. But that brought up some questions for us, specifically, what should happen if you take a prodrug and you give it to a bunch of cell lines that don't have livers? Nothing should happen. So how on earth did they identify sensitive and resistant cell lines in the first place? So with that as a question, we went back and we checked and we said, oh, well, actually the cell lines that they're using are sensitive and resistant, but to pemetrexed. They got the drug wrong, but they got great results, so who cares? So we reported this to the Lancet Oncology saying, here are some problems. And the Lancet Oncology got back to us and said, well, you appear to be arguing about statistical issues, to which, as everyone knows, there are no right or wrong answers. So as a result, we don't see the need to publish your objection. And we took it because we had no idea how to respond. And we said, okay, and we moved on to the next thing. And the next thing that happened was that some colleagues of ours who were working with gliomas, brain tumors, said, hey, actually that group is now talking about temozolomide, which is a drug we use a lot. Here's the heat map they came up with. Can you check this and see if you can understand how this came up with? And we said, okay, we think we understand the approach now. Sure, so we took the data and we tried to match it, and we tried for weeks and we couldn't get this to work. We couldn't match this heat map, we couldn't get the gene list, and we said, what's going on? So given that we couldn't get it to work initially, we said, we must have missed something in some of the earlier papers. So let's go back at those and look at those earlier papers more closely. And as we did, we looked in particular at one of the ones dealing with cisplatin and pemetrexed. And we looked at the heat map for cisplatin. And we looked at the heat map nominally for temozolomide. And we looked back and forth and back and forth, and we said, you know what? Something's wrong here. One of these two is wrong because they're exactly the same. We don't know which one, but we're certain one of them is. So we sent a note about this to the editors of both of these journals and said, one of these is wrong, we'll let you figure out which one. And indeed, the editor of CCR got back to us shortly thereafter saying, oh, the authors made a goof, they supplied the wrong figure, they will supply a fix, no need to worry about it. And indeed, a few weeks later, the authors supplied a new heat map and a new gene list, which did not agree with each other, and neither of which agreed with the text. So this paper about temozolomide, drug A, has achieved the neat hat trick of having in it a heat map for drug B and a gene list for drug C, and we can verify the information for none of them. So that's a neat trick. Now, all of this going back and forth, that actually took a few years trying to figure out what was going on. We were doing this because people were excited at MD Anderson, we'd like to help, but the reason we really care came up about this time. And that's when we got contacted by some acquaintances who said, are you aware that they've already started using this approach to guide how patients are treated at their institution? Specifically, this is at Duke University. And we said, no, we weren't aware of that. And we went back and we looked at the clinical trials in question, and we saw that in some of these clinical trials, not only are they guiding them, some of these are using drugs for which, based on our examination of the data, they have the sensitive and resistant labels reversed. We were very upset. So we said, okay, let's write it all up. Let's write up everything we got, put it together in a single paper, and let's see if we can get it in somewhere. So we sent it off to the editor of a prominent biological journal first to see what he had to say. And his comment came back was, well, your story's too negative. Can you fix that? And we said, you know what, we really don't think we can. So we sent it off to the Annals of Applied Statistics because, well, we figured everybody would still see it there. And there, fortunately, the response was a bit different. We got a note from the editor the following week saying, you know what, we've simply decided to accept the paper as is. It's posted online now, which means I send lots more papers there. That's a good thing. But it also meant that since the paper was now in the literature, we could cite it to the press. And just a few weeks later, the Cancer Letter, citing our stuff, ran a headline story saying, biostatisticians at MD Anderson are alleging the potential for patient harm in clinical trials being run at Duke University. And the National Cancer Institute, unbeknownst to us, sent separate notes to Duke's Institutional Review Board saying, we've checked this paper and we see the same types of mistakes in grant applications this group is submitting here. Please check this and reassure us. And Duke, not wanting to piss off the NCI, said, you know what, we're going to suspend these trials, suspend enrollment, and we're going to invite in a new group of experts to look at the data and see if everything makes sense. And we breathed a sigh of relief. We were really upset that it got this far in the first place, but given that it had, we could deal with an investigation. So we said, what's going to happen next? And while we waited for the report, the authors at Duke posted brand new data to their website. The data that they posted was, they said, these are the test data that we use to prove that our signatures for cisplatin and pemetrexed work. There are 59 samples from a larger public data set. Here's which samples we used, and here's the labels we used. We said, great, let's check it. Let's see if we can match it. So we tried to match the new data. And when we checked the data, we tried these correlations. And what you should be seeing in this plot is, if all of the 59 samples are what they say they are, you should see 59 red squares, perfect matches, all on top of this blue line, where the name agrees with what the numbers are. None of those red dots are on the blue line. Actually, there's only 43 red dots in this square at all. That says that for those 43 samples, we can perfectly match the numbers to raw data, and the sample isn't what they say it is. They got the name wrong, and hence they got the clinical annotation wrong. What about these 16 samples? For these 16 samples, there's no red square, because for these 16 samples, they've already scrambled the gene names so badly that these aren't samples of anything anymore. But what we can tell you is that every single one of the validation samples was wrong 2 years after they'd been started clinical trials using these drugs. And we reported this to Duke and to the National Cancer Institute and waited to see what would happen next. And what happened next was that Duke said that they were going to restart the clinical trials, because their independent investigation had strengthened their confidence in this evolving approach to personalized cancer treatment. We said, that's surprising. And we said, will you show us the data justifying this decision? And Duke said no. Duke said the reviewers approved of our sharing the report with the NCI, but we consider it a confidential document, and a future paper will explain the methods that we're using. So we're going to go ahead and treat patients this way now, but we're going to explain what this makes sense later. And this disturbed us, but we actually looked more closely at what Duke actually said. While the reviewers approved of our sharing the report with the NCI, and we paused there because the NCI, that gave us an in. The NCI is the National Cancer Institute. It's a federal agency, and as such, it is subject to the provisions of the Freedom of Information Act. So several months later, we got a copy of this report, not from Duke, but from the NCI pursuant to a FOIA request. And when we looked at the expert's report, we said, this does not justify restarting trials. What the hell's going on? And Duke had no comment. And we looked at that and we said, is there anything else we can do? And we were just sitting around feeling rather depressed because we didn't really see what we could. And then I got a call one evening in my office, and the guy said, hi, I'm a lawyer. Here's some information my client thinks you should have. And I talked with him for a while and I said, that's really interesting. Here's a reporter you should talk with. And I called Paul Gorbler of the Cancer Letter. And Paul talked with him, and Paul gave me a call about an hour later, and said, wow, I'm going to have fun. Why was Paul going to have fun? Well, 2 weeks later, he got to write a story that said one of the Duke investigators used to claim on his grant applications and on his CV that he was a Rhodes Scholar, but the Rhodes Foundation disagreed. So that was a bluff. It wasn't right. So now we had evidence that, okay, the science, not only wasn't the science right, the CV wasn't right. Now, this story appeared on Friday. I got a copy. I spammed all of my friends and said, I know some of you have been talking about writing a letter. Now is the best time you're ever going to have. And so a bunch of biostatisticians wrote a letter to Harold Varmus, head of the NCI, saying, would you please resuspend these trials until we can justify the science? Varmus called Duke and said, you're going to suspend these, aren't you? They said yes. And they suspended him. He contacted the Institute of Medicine and said, will you run an investigation? The IOM said, yes, we'll start in December. But before we got to December, there were a few new events. In October, Joe Nevins, the senior author on most of these papers, sent a note to his co-authors saying, you know what, we may have to pull one of the papers. Specifically, we may have to pull the paper dealing with cisplatin and pemetrexed because closer examination of our database shows that 43 of our 59 samples were mislabeled and we can't match the other 16 samples to raw source data, which is what we told them the previous November before they enrolled more patients. A few weeks later, Duke officially terminated the clinical trials and announced that in retrospect, these trials should never have been run. And then a few weeks later, other retractions happened, the other author resigned from Duke, and a cascade of things began. Here's some of the cascade. There were 100+ patients in these trials, lawsuits filed. There was a misconduct investigation that ran for 5 years in pulling 10 or more full retractions from journals like New England Journal, Nature Medicine, JAMA, otherwise high-profile papers. It hit the front page of the New York Times, it hit 60 Minutes, and actually, since I'm vain, we're gonna try running this 60-minute clip and we're gonna see if we can see what happens there. So.
Dr. Nevins discovered that when the underlying data disproved Dr. Pote's theory, the data were changed.
It became clear that there was no explanation other than there was a manipulation of the data, a manipulation of somebody's credentials, and a manipulation of a lot of people's trust.
Manipulated data. These were not errors.
That's correct. It simply couldn't be random. It simply couldn't be inadvertent. It had to been based on a desire to make something work.
Is it a close call or is it abundantly clear that the data were fabricated?
Abundantly clear.
It was abundantly clear that the data were fabricated 5 years after clinical trials began. It shouldn't happen quite that late in the game. So part of the fun thing here is that here's another question for you in the audience. What would you do? Because something that only came out 5 years after this crash down was that there was actually a student in Anil Pote's lab, one of the labs where this was going on, who at the time got very worried, and he wrote a note to Duke saying, there are problems here. Can we fix this? We only found out about this during the course of discovery in the lawsuits. It came out later, but the reason it didn't happen earlier is because this got mishandled badly within Duke. Specifically, he went to Duke Dean saying, I have this problem. And they said, you're right, that's a problem. You should talk with Joe Nevins, the senior author on the papers, somebody who has a distinct conflict of interest. And as a result of that direction, these criticisms never saw the light of day until years later. So that's a bit of a problem. But it's also something to think about as you say, would I be able to do something, or how would I handle it if something I was working on turned out to be wrong? Now, some quick observations. This story that I just told you about it's a worst case scenario. We don't have this many mistakes happen all at once. But I can tell you that we were able to recognize the individual types of mistakes, label errors, things like that, because we'd seen each and every one of those types of mistakes in data that we get at MD Anderson. It's not because MD Anderson's a bad place. MD Anderson's actually a great research institution. It's that these are simple mistakes and they're easy to make, particularly if you're working with your data in Excel. It's easy to swap columns, swap rows, and things like that. But if we have access to all the data, we can say, oh, well, wait a minute, that looks weird. Let's check, oh, here's what happened, there was a swap, and we can fix it. But what happens is, if we then have a simple mix, and we then go to publish something and we say, oh, well, the journal won't give us space to publish everything. We'll just publish the final results, not anything in between. Then the nature of that mistake has just become hidden, and we become worried that actually simple errors may be more common in the literature than we'd really like to see. And actually checking the literature shows that this is indeed the case, unfortunately. There have been a few different papers. Probably the most high profile one recently was from Baggerly and Ellis, who wrote in Nature, and this is in 2012, and they said, working with groups at Amgen, we got together with the authors of a whole bunch of breakthrough clinical papers and said, can we try to replicate it with you because we want to commercialize it? Working with the authors of the initial papers, the authors were able to replicate these results in 6 out of 53 studies. And that scared the authors, and it scared the NIH, which is funding all this stuff, saying, we don't want to do that. Let's do it better. Furthermore, how much of this stuff is being wasted? So here, let's give you some dollar amounts. Here's the NIH budget. We think about half of that is being wasted because of irreproducibility. If we break that down, let's rephrase these. Biological reagents and reference materials. This is, did you get the labels right? Study design. Did you avoid confounding? Data analysis and reporting. Did you tell us what you did and show us the code? That's 90% of $28 billion lost due to simple mistakes that we can fix if we're aware of them. Now, we were asked what we thought should be required in a whole bunch of these studies. And actually, as we started looking at it, particularly when you start getting with complex data, we need the data, we need the description, and we need the code. Because we've now passed the point in data mining where verbal descriptions are adequate anymore. And actually, there's another way of putting this. So I'm going to try telling you one more way of thinking about what we need. I want it now, specifically when the paper is published, because that is the point when journals and the rest of us have leverage to say, give it to us. We can make you do it. Now, that's all told most of my story. I do have to tell you, though, that even after all this, I'm actually an optimist. I think things are going to get better. And the reason I think this is that having told this story a lot and having worked on this for many years, I've seen the trend of the quality of the data that is emerging. And I'm seeing the development of tools to make it easier to do it right. So I've listed some of the tools here. I've listed the actions of journals. Jenna mentioned the fact that the NCI has now initiated a rigor and reproducibility initiative. That was in 2016. That governs things that they fund. And there's things like Project Tenure and the Center for Open Science, which are also telling you how should we do it right. Here's some places to learn more if you want to read more about it. I have to acknowledge I didn't do all this myself, so here's some people who helped contribute, including Kevin Coombs. If you want to read more, you can read the Annals of Applied Statistics paper, because there's only 3 formulas in it, and they're all wrong. And finally, I have one last thing to do, which is to thank you guys for having put up with me for this talk, because that's really what I've got. And that's what I've got. Thank you very much. Time for questions.
Thank you so much. That was phenomenal. We have one comment on the Q&A, which is just, this is terrifying. And I would have to agree. If there are any other comments or questions coming from the audience, please feel free to type them in the Q&A box. I'll give you a second in case you have one sort of queued up, waiting to send. Otherwise, I have my list of questions that I can also, let me put my video back on. You can all see me.
Okay.
Okay, I will go ahead and ask my first question, but please feel free to chime in if you have any other questions for those who are still in the audience. So one of the things that struck me when I was looking at your slides is that it seems like there are actions that were taken by various entities at the conclusion of this. So for example, the NIH, it seems, individual scientists who hear about the story are interested in maybe learning more tools to deal with reproducibility. But what specific changes have you seen for journals aside from maybe pushes for reproducibility? Have there been any, I guess I'm thinking more on the side of after a paper is published, when these issues are identified, what would this process go differently if you were to write write these cancer journals again?
So I'm going to give you a two-part answer to that. The first bit is that most of the fixes that the journals have put in place already have to deal with what happens before the paper is finally accepted for publication. They're saying, we want to see this, we want to see, here's some things you can supply. As a reviewer, if I'm reviewing a paper, one of the things I can look for, and the journals will back me up on this now, is I'll say, oh, by the way, as I read through the paper, is there a link in the paper saying the data's here, the code's here? And if there's not, I'll say I need to see it before this is published. If we had to do this again, and this has actually come up for us, and sort of scary, actually one of the things that I would do differently that I didn't think to do the first time around is, quite frankly, I would call the editors, because many of the journal editors don't feel comfortable judging the science because they don't always think that they're qualified to do so, and they know that some of the objections they get are, quite frankly, from cranks. So the purpose of calling the editor is simply to say, hi, I understand you don't want to take this piece. Fine. Can you give me 5 minutes to let me discuss with you why I think it's really important in simple language? This isn't an arcane issue. It's the fact that they got labels wrong. And in addition to that, as I make my case, to probably persuade you that I'm not a kook. And this is something that I learned to do only after all this came down, because several journal editors approached me and said, why didn't you call us? And I'll just tell you that I didn't think of it at the time. So that's why I didn't do it. But that's what I would do differently. Okay?
That's a great suggestion. We have a couple of questions that came in. So the first one that came in is, how do we change the academic reward system, specifically tenure and promotion, to incentivize the opening of data sets earlier in the process?
Well, to get the incentivize opening data sets earlier, I'm not really sure on that one. To incentivize the sharing of data for many of the ones that are funded by NCI or whatever, there is now a requirement that the data generated as part of this grant must be published by the end of the grant. And for publication, there's typically also some embargo that once you submit to a journal, they expect you to have submitted the journal to a public repository at that time. So at that point, you have about an hour, a year, not an hour, to get your paper finalized and accepted, and that's it. And then it will become public. So the other thing that the NCI and granting agencies are doing is, we have talked with them about, can we also look at citation rates for data sets, not papers? Don't know how precisely to do it. We'd love to do it, but that's an idea that we've tossed at the funders, but it's really the funders who are going to have to address this, unfortunately.
Yeah.
Okay.
I'm going to summarize. There are two questions that sort of came in on sort of standards for reproducibility. Specifically, the first question has to do with the differences by field. So if you're doing a relatively simple, say, linear regression analysis, maybe it's really easy to put all of the documentation necessary to reproduce it. But what information would you need for more complex studies, and how could you standardize that, say, for genomics data?
There is indeed a lot of field-specific constraints. And indeed, that's why for genomics, there are guidelines for RNA data, guidelines for genomic data, guidelines for sequence data. So it's all separate. But as a rough rule of thumb, have you supplied enough data in anonymized form and enough code that somebody else can run that code and generate all the figures in your paper? That's really sort of a bottom line thing, is, can we at least see what you're doing? And that's the rough and ready thing that I would say across field. Before that, beyond that, I'd have to get more specific, and that's a bit too granular for me right now.
Yeah, so the idea is that, you could maybe not have a standard, but as long as you can make sure you can get from the data to the results, regardless of how complex the study is, then you've sort of made progress.
Yeah.
The next question is whether you ever received an apology from the journals for being brushed off, and were you ever thanked by Duke for all the work that you put forth?
Two things. After all this broke down, I got a call from the editor of the Journal of Clinical Oncology. And he said, it is not the policy of our journal to keep copies of papers we have decided not to publish. However, it has come to our attention that certain objections you raised 3 years ago were very likely correct. Would you be willing to resubmit? We bit our tongues and resubmitted. This was about cisplatin and pemetrexed. That paper was retracted the following week. Our objections were never published and never will be. But JCO has apologized, and that was there. After all this came down, Duke did institute new policies at their institution, and the people leading that effort got in touch with us and tried to work with us very extensively. They tried to fix things, and they made a good faith effort at that time. Unfortunately, that was a bit after, but yes, they did make a good faith effort at that time. There is an issue here about how long the government takes to move, which is that as a result of all this, actually, the government put additional funding restrictions on Duke 2 years ago for something that actually crashed down closer to 8 or 9 years ago. So there's lots of inertia. If you piss off one group, it will come around eventually. Okay.
That's a good thing. So it seems like the last comment is a bit more about maybe perceiving these changes for reproducibility less as requirements and more as guides to help authors advance science. And I guess whatever perspective you could offer on that idea.
Actually, the general thing here is that the general principle I want the authors to be having is that if somebody wants to double check my work, it should be easy for them to do. Have I supplied enough to make it at least to first order easy for them to do? Or if I can't, have I provided an explanation for why not? This comes up a lot, but so if people are working from that principle, and we've contacted people who've written papers where we're trying to track down details, most of them who've supplied at least some of the data, who've made a good faith effort, if we haven't been able to build it right there, they've been able to address it fairly quickly. So that's really the mindset that I'm trying to get people to adopt. I can't get, again, too specific, but if you're going in with that goal in mind, if you say it should be easy for somebody else to do, then you're not going to get too far off at the end of the day. So that's the bottom line.
That's great. Well, I think that's all the time we have for questions. So I want to thank you so much for a wonderful talk, and thank you all for being in the audience. And I think we'll end here.
Okay, thank you very much.
Thank you.